Howdy, my name is Jazlyn Gonzalez and I am part of group number 6 with Andres Garza. Today we will be going over biochemical tests 4. In this lab, we use 3 tests to tell common bacteria apart by their enzymes and hemolysis patterns, which are the urea test, CAMP test, and the coagulase test. On the urea agar, urease-positive organisms split urea to ammonia, which increases the pH and turns the medium pink, while urease-negative strains leave the tube its original color. For the CAMP test, we made a streak of Staphylococcus aureus down the center, Streptococcus pyogenes above, and Streptococcus agalactiae below. A positive CAMP reaction is seen by the arrowhead-shaped enhanced beta hemolysis. Finally, the coagulase test uses rabbit plasma in a tube where clot formation indicated a positive result, while liquid means negative. These tests together help distinguish bacteria by their unique enzymatic activities and pathogenic traits. Now for our materials. For culture, we used Escherichia coli, Proteus vulgaris, Staphylococcus aureus, Staphylococcus epidermidis, Streptococcus agalactiae, Streptococcus pyogenes, and our unknowns number 136 and number 276. For media, we used 4 urea tubes, 1 blood agar plate, and 2 rabbit plasma tubes. And for our equipment, we used an inoculating needle, an inoculating loop, and a butane tank as our aseptic heat source. Now for our procedure. For the urea test, we streaked each urea agar tube with their respective culture of E. coli P. vulgaris or our unknown number 136 and number 276. The tubes were then incubated and viewed for results during the next class time. For our CAMP test, on our blood agar plate, we did 1 streak of S. aureus down the center, followed by 3 streaks of S. pyogenes above that line, and 3 streaks of S. agalactiae on the bottom of the center line. It was then incubated and viewed for results the next class time. For our coagulase test, 2 tubes with 0.5 milliliters of rabbit plasma were inoculated with S. epidermidis and S. aureus. The tubes were then incubated and viewed for results.
Now moving on to the results and observations for the urea test. So for the urea test, urea agar tubes were inoculated with several organisms, including E. coli, P. vulgaris, unknown 136, and unknown 276. After incubation, only unknown 136 and 276 showed a clear color change from a yellow, orangish to a pink color on top. This indicated a positive urea test, which means that these 2 unknown organisms produce the enzyme urease. Urease essentially breaks down urea into ammonia and carbon dioxide, raising the pH of the medium and causing the pink color to appear. So these tubes that contained E. coli and P. vulgaris did not change color and remained yellow, a yellowish orange. So they were considered urease negative in this experiment. Unknown 276 was also identified as gram-negative from our previous labs, and when combined with its positive urea result, this suggests that it may share more characteristics with gram-negative urease producing bacteria. Now for our results and observations with the CAMP test. A blood agar plate was used for Streptococcus agalactiae, Streptococcus pyogenes, and Staphylococcus aureus. On the plate, Streptococcus agalactiae was streaked on the far left side, whereas S. aureus was in the middle with 1 streak, and S. pyogenes was 3 streaks on the far right. So now, after incubation, Streptococcus agalactiae produced an arrowhead-shaped zone of enhanced hemolysis near the central Staphylococcus aureus streak. This pattern indicated a positive CAMP reaction, which is characteristics of S. agalactiae. On the far right, Streptococcus pyogenes did not show an arrowhead pattern and remained CAMP negative. This test demonstrated that the synergistic effect between the CAMP factor produced by S. agalactiae and hemolysin produced by S. aureus. And it also helped confirm the identity of S. agalactiae on the plates. As for the results and observations with the coagulase test, rapid plasma tubes were inoculated with Staphylococcus aureus and Staphylococcus epidermis. These tubes were incubated at 37 degrees Celsius, and after incubation, the tube containing Staphylococcus aureus formed a visible clot. In person, you can see it, but on the picture, it's not as clear. But essentially, this indicated a coagulase positive result. And coagulase is an enzyme that converts fibrinogen into fibrin, causing the plasma to clot and allowing the bacteria to form a protective barrier against host defenses. So, in contrast, the tube containing Staphylococcus epidermis remained liquid, indicating a coagulase negative test or negative result. Sorry. This difference in coagulase activity helps distinguish the more pathogenic S. aureus from the less virulent S. epidermis and other coagulase negative Staphylococcus species. So in conclusion, the biochemical tests performed in this lab helped identify enzyme activity and possible bacterial characteristics of unknown samples. In the urea hydrolysis test, both unknown 276 and unknown 136 produced a pink color change, indicating positive urease activity and the ability to break down urea into ammonia and carbon dioxide, which raised the pH of the medium. Since unknown 276 was also determined to be gram-negative, this result aligns with bacteria such as the Proteus species, which are known for causing urease production. In contrast, E. coli remained negative, showing no color change. And as for the CAMP test, it confirmed that Streptococcus agalactae as positive through the formation of an arrowhead-shaped zone of hemolysis with Staphylococcus aureus, while S. pyogenes was negative. The coagulase test showed that Staphylococcus aureus was coagulase positive, forming a clot and forming the pathogenic potential, while S. epidermis remained negative. Overall, the results demonstrated how enzyme-based biochemical tests, along with gram staining, are essential tools in differentiating bacterial species and understanding their physiological and pathogenic traits. Now for our references, this is what we used to complete our lab report. And finally, this concludes our video lab report presentation. Thank you all for listening.